Question number 1 is specifically asking about the difference between chromosomes genes and DNA. The fourth part of the question is actually asking how all of these relate to the process of protein synthesis. Which of course involves transcription and translation. So the question itself is a little bit more complex than it looks. This whole area about transcription and translation has everything to do with the genes that are being translated, and the DNA is obviously making up those genes, and all that DNA in the form of genes is packed tightly together with chromosomes. So to get more specific, we'll get to the next slide and look at it a little closer. But before we do, I'll try drawing. You guys may remember up on the board that we drew pictures of chromosomes. So for example, this would be our number 1, according to what we see up on my number 1 up here. Those are my chromosomes. And located on my chromosomes, we have sequences of DNA. And those little sequences of DNA are our genes. Human beings, as we remember, have 46 chromosomes, or 2n equals 46. N, which would be our haploid cells, equal 23. Now for a little better of a picture, you can see the chromosomes right here, circling it in white for some unknown reason. Circling it now in pink, there's our chromosomes, and sequences of chromosomes are actually made up of genes, and those genes themselves are in fact made out of repeating sequences of adenines and thymines, A's to T's and G's to C's, which of course are our base pairs, and that makes up our strands of DNA. How, though, does any of that relate to protein synthesis? Oopsies. So don't waste too much time trying to draw this. I'm going to put this picture in right away. And in the upper left-hand corner here we see our DNA. The double helix, we don't see it in the chromosome form, where we have our classic 2X sort of shape. We're looking at it in the DNA form because the DNA is going to be transcribed into RNA, and you see that in that purple line or green line that's coming out through the nuclear pores. So that's our mRNA. Keep in mind that transcription also makes tRNA molecules that you see right there in yellow, and it also makes rRNA, which is that red blob there and there. So it's important for you to remember that you've got DNA molecules. So there's our DNA molecules right there. Alright, again 2n equals 46 if it's a human being. And then that DNA is transcribed into an RNA molecule in 3 different types. You've got messenger RNA, which you see here in the blue line. You have transfer RNA, which look like these little yellow T's. And then you have ribosomal RNA, which makes the ribosomes. So the first part, again, is transcription. Transcription takes place inside the nucleus, and then that RNA comes out of the nucleus and into the cytoplasm, where the mRNA the ribosomes, and the tRNA are all used to construct proteins. So back to the original part of the question, what do proteins, or what do chromosomes, genes, and DNA have to do with protein synthesis? That sort of sums it up.